back to biology. We're going to start our second semester coursework today, so get excited because we're going to focus on the smaller molecules of life. Our focus today is primarily on DNA structure and its function. So we're going to learn a little bit about what DNA is, how it works, and what does it code for in the body. We know that DNA is the code for life, but how does this work and how is it arranged so that it is efficiently used inside of the cell? Those are just a couple of things we're going to look at today as we explore more on DNA structure and function. So hopefully by the end of this video lecture, you'll be able to understand the discoveries that led to the discovery of genetic material and its structure. The history behind how DNA was found is very interesting. We're only going to cover just a brief portion of it. But if you do a little bit more research, you're going to find some very, very interesting and controversial studies in order to find DNA, discover it, and learn its purpose. And hopefully you'll also be able to explain the basic structure of DNA because the structure of DNA determines its function. All right, let's get started. The concept that traits have been inherited and passed on from generation to generation has been around since the ancient Greeks. But it really wasn't until 1952 that Hershey and Chase actually proved that DNA, which had been known for some time, that there is a genetic material known as DNA, that it actually codes for proteins inside of the body, that DNA structure that's found inside every single cell inside the nucleus codes for the proteins that allow our bodies to function. It was 1 year later that the famous duo of Watson and Crick determined that DNA was in the shape of a double helix structure. Now this is very important to know the structure because it helps us understand how do we make those proteins, how do our cells make proteins, how does DNA make copies of itself, and how does this in essence code for our entire bodies. So the structure is very important because it helps us determine what the function of that particular molecule is. So Watson and Crick are very, very famous for their discovery in 1953. This discovery, which occurred in England, wasn't just found on their own. They had help from many other scientists to help them discover, but they do get most of the credit. All right, and this double helix structure is what we know today as the DNA molecule. So what specifically is the DNA molecule composed of? Because it is a very large molecule and structure that determines the proteins that are made inside of our cells and bodies. DNA is composed of monomers. DNA is the polymer. The monomers are known as nucleotides, and each nucleotide contains a sugar, a phosphate, and a nitrogen base. The nitrogen bases are the code. The phosphates and the sugar make up the backbone of the DNA molecule. So if you picture it as kind of a twisted ladder is the term that we use in biology class. The phosphate and the sugar are the outside parts of the ladders that you would hang on to as you go up, and the rungs of the ladders, or the things that you would step on, would be the nitrogen bases. Now, there are two different types of nucleic acids that we're going to look at in this course, DNA and RNA. DNA contains deoxyribose sugar, which is why it's called DNA, because it's different. The sugars are different between DNA and RNA. That's what allows them to have different names. RNA simply contains ribose sugar. All right, so there's the key difference between DNA and RNA is the sugar molecule that exists within the DNA structure. So keep in mind those nucleotides contain a sugar, a phosphate, and a nitrogen base. The sugar differs depending upon whether you're looking at DNA or RNA, and the nitrogen bases determine the sequence that determines the code that determines the protein. All right, there's a lot of cause and effect here when we look at the structure of DNA. Speaking of those nitrogen bases, there are only 4 different nitrogen bases that are found in DNA: adenine, thymine, guanine, and cytosine. These 4 nitrogen bases repeat themselves within the sequence of DNA to make a long code that codes for proteins in the body. The big difference with RNA, however, is that uracil replaces thymine. So there's a difference there. There is no thymine in RNA. However, there is uracil. In 1950, a man by the last name of Chargaff noticed that the amount of adenine in DNA is similar to the amount of thymine. So what this lets us know is that A will always bond with T. Remember, DNA has two sides to it. It's not just a one-sided figure, but there is a double helix. There are two strands of DNA. The A binds with the T, and the G binds with the C. So we always know that the amount of T is the same as the amount of A, and the amount of C is the same as the amount of G. The easiest way I remember this is to think about where you go to school. You go to school at CG, so you'll always know that A bonds with T and C bonds with G every single time. All right? So when we pair up each individual strand of DNA, we'll see that A will always bond with T, C always bonds with G. So the amount of A is equal to the amount of T, and the amount of G is equal to the amount of C. We may do a problem with this a little bit later on today. So again, you look at the DNA structure. DNA is in the form of a double helix. Here on this picture to the right, it has been flattened out to get a better understanding of the structure itself. But you see that there are two strands to it, A binding with T, C binding with G, and that they're held together through what's called hydrogen bonds. And those are the same things we talked about in the biochemistry unit. Those hydrogen bonds hold things together just like they hold water molecules together. Those hydrogen bonds hold the A and T and C and G together. Now they can be broken apart if need be for processes like DNA replication and protein synthesis, which we will talk about a little bit later on in the unit. The backbone of the latter is made up of the sugar and the phosphate. So you see the phosphate group and the sugar group that make up the backbone of the latter. And the nitrogen bases make up the steps of the latter. But again, you can see that A will always go with T and C always goes with G within this DNA structure. You just have to imagine that this picture, three-dimensionally, would be twisted. You would just twist that or rotate it. All right? One thing that's interesting, and we'll learn a little bit more about this when we get into DNA replication because it has a big impact there. But each strand has what's called orientation. And the way the sugars are oriented on the strand determines the orientation of the DNA. So one strand of the DNA runs what we call 5' to 3', and the other runs from 3' to 5'. So you can see the sugars that are facing up, all right, start at the 3' end and go up to the 5'. However, when we look at the opposite strand, the sugars are running the opposite direction. So the 3' end is on the opposite side of the DNA molecule. So if one end of the DNA strand is 5', the complementary strand, or the strand opposite it, will be 3'. All right, they'll always be opposite of one another. You'll never see two 5' ends together or two 3' ends together. Strands will always run in opposite directions. Now, DNA doesn't just exist in the cell as this double helix molecule. It's actually tightly compacted into what's called a chromosome. Specifically, eukaryotic DNA is arranged into chromosomes, and these are tightly coiled DNA that's wrapped around the histones. If we unraveled DNA, all right, it would be entirely too long to be able to fit inside of a cell. So much like a hose, it has to be wrapped tightly in order to fit in the space that's required. The DNA is tightly wrapped around proteins called histones, and these histones act kind of like the wheel that the hose wraps around. Prokaryotic DNA is a little bit different because it's simply found in a circle known as a plasmid. Remember that prokaryotic cells are not nearly as complex as eukaryotic cells, so they don't have quite as many functions or features as a eukaryotic cell would. So hopefully you understand a little bit about the discoveries that led to the discovery of the genetic material and its structure, and that you're able to explain the basic structure of DNA. Hopefully you understand this quite well. We'll do a little more work with this later on, so don't forget to take your video quiz, and I'll see you next time, guys. Have a great day.